episode of the BioHub. Today, we're thrilled to have Dr. Chris Haqq, Chief Medical Officer and Head of Research and Development at Elicio Therapeutics. Chris is an expert in immuno-oncology with over 20 years of experience in the biotech industry. He has a remarkable track record in leading innovative therapies from concept to clinical stages, including his pivotal role in developing groundbreaking T-cell therapies at Atara Biotherapeutics. In this episode, we'll explore Elicio's unique approach to cancer immunotherapy, Chris's insights on the current biotech landscape, and also what it takes to lead in such a challenging field. It's an absolute pleasure to have you, Chris. How are you?
Good. Thanks so much, Max. It's a pleasure to be with you today.
No, it's great to have you. Seriously. So, Chris, can we just start by you sharing the story behind Elicio and also what inspired you to join the company?
You know, I have a pretty interesting family story there, as my dad was diagnosed with a KRAS mutated colorectal cancer just before I joined. And I was struck at that time how the drugs he was prescribed were actually the same ones that I'd learned about all through my training, even in medical school and when I became an oncologist. And with his passing, I saw firsthand just how much impact we could have by developing better agents that could harness the immune system for GI cancers. I had also had a pretty interesting educational experience there, as my MD and PhD training was at a joint program at Harvard and MIT called the Health Sciences and Technology program. And the technology we're developing at Elicio, the amplifier technology that I'll kind of call AMP throughout our discussion, was developed in Daryl Irvine's lab at MIT. And it's just a perfect example of the goals of the Health Sciences and Technology program, which try to bring together engineering approaches to solve important health issues. And the idea of the AMP program is to engineer drugs to ride piggyback on albumin, which goes naturally into lymph nodes. And the lymph nodes function as kind of the brain center for the immune response. And when I heard that Elicio had been formed to develop the AMP technology to target the very RAS mutations that affected my dad, and that they were looking for an oncologist to help them make that really key transition from the bench to the bedside, it was just such a great fit for my background. I come from the adoptive T-cell therapy world, and it fit perfectly with my desire to develop cancer immunotherapy for GI cancers.
I think that's always amazing when it works out like that you know you get to work on something so close to your heart, and I think that's often the case in biotech. I think it's really interesting because it just motivates you but so much more as well.
Yeah, you feel great waking up every morning knowing that what you're doing could affect so many people, similar to the people that are the loved ones in your own family.
Yeah, let's just talk about that AMP platform a little bit more. What excites you most about it, and also how does it harness the natural power of the lymph nodes to enhance immune responses against tumors?
Yeah, for a translational oncologist like myself, it's so energizing to be a pioneer. I'm leading the first clinical studies in humans for a whole new approach to immunotherapy. Here we know that older approaches produced limited numbers of T cells, especially for the single amino acid mutations that our whole field desires to target for these cancer driver oncogenes like KRAS. And the historical approaches in mice were usually not able to generate both of the key T cell types. So we typically think of the helper CD4 cells as well as the killer CD8s. And when those were made, often lower levels of their effectors, like cytokines that can broaden out the immune response, were seen. So by contrast, with the AMP platform we're developing, we could see that through the engagement inside the lymph node, we got to very high levels of both types of cells. In fact, in 59% of the patients in our phase 1 publication, which came out in January in Nature Medicine, we saw both types of T cells respond, and that those T cells were really active. They were able to make the key cytokine mediators, and the immune responses then really correlated with what happened for the patients as they were followed. We looked at the median relapse-free survival times of the patients participating in our phase 1 and saw that the patients with the above median T cell responses had a relapse-free time that wasn't reached, compared with only 4 months in the group that had low or no T cell responses. And that comparison gave a hazard ratio of 0.14, a p-value of 0.0167. So it's pretty exciting in a phase 1 investigation to see the correlations of the mechanism of action with the outcomes like that. And that really got me excited to take the next steps and enter ELI-002 into the later phase of clinical development.
Yeah, I think that's all incredible. And I'm sorry, but I obviously have to ask this question. And there is a little bit of skepticism, and there's also recent failures in cancer vaccines, such as Gritstone, for example. How does Elicio aim to overcome the same challenges these guys had and also stand out in the field?
Yeah, I'm glad you asked that. So I think in the past, some clinical trials have made use of novel endpoints in development. And our strategy to overcome challenges in the field is to generate randomized clinical trial data using the most traditional oncology endpoints, because that's what has actually led to the marketing approvals that we have in hand for our field with cancer vaccines. For example, relapse-free survival, like we were just discussing for the phase 1 correlations with our ELI-002. That was used for BCG and bladder cancer, which is one of our mainstays. And overall survival, another more traditional oncology endpoint, was used for the approval of Provenge, a cancer vaccine in prostate cancer. It's just a couple of examples that come to mind.
Our randomized phase 2 Amplify-7P trial in pancreatic cancer began its enrollment in January and is anticipated to complete accrual in the fourth quarter of this year, 2024. Amplify-7P has the primary endpoint of disease-free survival and will also allow us to collect the initial information on overall survival. In parallel, we're going to seek regulators' advice so we can start planning for phase 3. And one really important design feature that I wanted to mention is the crossover design of the Amplify-7P study. We're using radiographic response to assess when patients who were initially assigned to the control group then cross over and receive ELI-002 after they've had a progression. Well, that'll allow us to measure the radiographic response in their metastatic tumor. And if the data in that crossover group are promising, that holds the potential to motivate future trials that use a radiographic response rate endpoint to broaden the group of patients who could benefit from ELI-002. Since only a subset today can have surgery, but virtually all patients receive treatment for metastatic disease with this terrible tumor type.
So I think this kind of clinical design, relying on the more traditional endpoints that have been able to support approvals in the past, is very, very important. And even though it sometimes requires a larger sample size and a longer time to read out, having those type of data can really serve the field, because they should be able, we anticipate, we believe, to be able to predict the outcomes for the later phase studies.
Is that where those struggles were with these other companies? You know, not necessarily using a traditional readout method, or they elsewhere as well? Was it like deeper engrossed in their science or their tech?
Yeah, I think to me, a lot of the challenges have come from clinical design. So I'm more comfortable going with the traditional endpoints here, and I'm not expert enough in the technologies to be able to compare all the differences there. But I will say that when we look at benchmarking, we often turn to applications of our AMP platform in the targeting of COVID-19. So I think there in mice, we can compare many platforms to the amphiphile and see that amphiphiles really stand out, generating both a high T cell response as well as in that application, a strong antibody humoral response as well. So the platform that we're working with has a lot of potential more broadly. So we use it to make T cells in the applications to oncology, but the same lymph node targeting would work well for the generating of an antibody response if we need that, for example, for an infectious disease application. So I think it does stand out when we look at preclinical data sets like that. But the good thing is, I think that there's so much innovation in our field, and there are several promising cancer vaccines coming along now that essentially aren't your grandfather's cancer vaccines. They are able to generate more robust numbers and functions of T cells.
Yeah, that's interesting. So just kind of pivoting a little bit to what the current market is like, and also some of the other struggles in the industry, which is tough fundraising environment. How has that affected you guys at Elicio? And also, what strategies are you using to ensure continued progress?
Yeah, this is a great and very timely question. Biotechnology funding is certainly challenging in today's environment. And for me, the most important thing is to have strong clinical data as early as it can be generated. During the pandemic, I made a really important choice when our manufacturing was ongoing in the year 2020. We had completed G12D and G12R, two of the mutations within the RAS oncogene, when the manufacturing facility availability was limited. And there were many demands on their time, for example, to address by making COVID vaccines. And rather than waiting for the unknown additional time that would have been required to make the full set of 7 mutations in KRAS that I had originally desired to take forward into the clinic, we decided to pivot and carry out the first trial with the 2 peptide amphiphiles that were in hand already. And by doing that, we got our clinical data a year earlier by taking that decision. Since the 2 mutations, the G12D and R, are very common in pancreatic and colorectal cancer, those ended up being the 2 groups of patients that participated in our phase 1. And we saw early evidence of anti-tumor effects in both of those GI tumor types. So I think that was a great decision. We published that 2 peptide formula phase 1 data in Nature Medicine in January. And that went on to generate a lot of investor awareness and media.
Right. So investors are really keen to see randomized data as soon as possible. And to meet that expectation, it's been a strong team effort at Elicio to parallel track the 7 peptide ELIO2 formulation through its own phase 1 trial and activate the randomized portion, the phase 2 portion of our study, to generate that key data as soon as possible. We're proud of the strong execution on the trial, with the first patient dosed in phase 2 in January and the anticipated completion of enrollment in the 4th quarter, with interim data expected in the 1st quarter of 2025.
So it's been a big challenge, but I think investors are very keen to see randomized data as soon as possible. So we've made a really strong team effort at Elicio to parallel track the newer 7 peptide formulation. That is what we anticipate to be the commercial formulation. And so it's gone through its own really expedited phase 1. And as I mentioned, we activated our randomized phase 2 this January to be able to generate that randomized data set as soon as we can. We're really proud that our team has had strong execution on the trial. And with the anticipated completion of enrollment later this year, that sets us up to have the interim data readout in the first quarter of 2025. But beyond that, I think it is worth noting that even though investors will always guide a company, and I think it is very good for a biotech company to focus, it is still really important when you're looking at a platform technology like we have, and when you're looking at a target like KRAS that's present across multiple different tumor types, that consideration is given to broadening and being creative in that. So one thing I've done is to work with academic leaders and also grant agencies to potentially conduct additional programs and additional trials. As an example of that, we have grant funding from the GI Research Foundation, and that allowed us to broaden our targeting to use the AMP platform to address driver oncogenes like BRAF and P53 that, alongside with KRAS, are the most commonly mutated across solid tumors. So I think a lot can be done to address the current fundraising landscape. And in a way, it's actually very good that there's so much attention and focus on generation of the key data that will really inform everyone of the potential of the new technology as early as you can.
Yeah, I think that's key. Like, if you look at it from an investor's perspective as well, they're looking for less risky investments in what we don't know what's going to happen in this market, and that just lessens the risk when you've got that clinical data to abide to. I'm also interested, like what it's like from a personal standpoint. Obviously, you've got previous investors who have come into the company, certain expectations of Elicio to keep going and bring this important technology through. What's it like when you're out there in the market looking for fundraising? Those from a personal standpoint, how do you deal with it?
Yeah, I think from a personal standpoint, you just have to remember that everyone really wants the same thing. So I think investors, regulators, patients, and advocacy groups are all really aligned. We want to take the resources that we have available, the patients' time in volunteering to participate in clinical trials, the investors' dollars that can fund the conduct of the studies, and take the most promising technologies into those trials. So I think doing those definitive endpoints early on, not being afraid to check a new technology up against the endpoints that are going to really, really tell you the outcome, that's important. Because I think that allows you to take only the most promising technologies forward. We've been very pleased to see the initial phase 1 data with our ELI-02 platform. I think it really is like a pipeline in a drug, in the sense that it can be frustrating that, for example, our first trial is in pancreatic cancer, but my dad died of colorectal. Boy, would I love to have a parallel trial ongoing in colorectal cancer? Of course. So as a translational oncologist, it's a little frustrating not to have all the funding in the world to be able to take forward multiple shots on goal. But I think that's where you can work with the grant agencies and collaborators to potentially be able to get things off the ground in additional tumor types like that. And then the data sets that you read out will generate additional interest. Having the randomized data in pancreatic cancer should be a stepping stone to generating the data. With positive data in one setting, it should be more easy to generate the support that's needed to broaden the development. So it is really exciting, really challenging to realize that an agent like this could be applied to so many tumor types and not have the funding to do all at the same time. But on the other hand, I think that focus is really good to generate definitive data as soon as you can. And in the end, that might end up being the fastest way to bring an agent like this out to the benefit of patients across the diversity of tumors that really need innovation.
Yeah. What are your thoughts on the biotech market more broadly, I suppose, in the realm of immuno-oncology? Where do you see immuno-oncology heading in the next few years? Do you see it growing? What do you think?
Yeah, I mean, I really look back on how the world looked in oncology during my training in the 1990s when I did my fellowship and joined as a junior faculty at UCSF. And it really is interesting to look back and think of the agents that struggled to get funded in that time. So I think if you look back in that era, a great example is Jim Allison's pioneering work in checkpoint inhibitors, which we've now seen translate, of course, into approvals across many tumor types where T cells are present that can be engaged by those checkpoint inhibitors. And at that time, it was very difficult to get grants funded in that area for something that now we look back and think of as, hey, that was a no-brainer to advance. So I think, where we don't see success with checkpoint inhibition is in those colder tumors where there aren't great T cell infiltrates when you look histologically at a section of the tumor under the microscope.
Mm-hmm.
And I think that's the gap that great new vaccine technologies like Elicio ZAMP come in, because the newer technologies are really able to prime new T cells to be born, as well as to amplify. We call our trials Amplify because they do such a good job with this technology of increasing the numbers of cells that might be present in too few number at the onset for a patient to have an effect on their cancer. So I think reversing the cold tumors to hot will then be able to synergize with some of the agents that we already have going, like checkpoint inhibitors. And I really do think that the current state is all monotherapy, and that we'll see combinations come down the road in the future as we become smarter about how to put together different immune agents, and that will really recapitulate the whole history of cancer treatment. I think back to, in my fellowship era, we had just started to have regimens like CHOP therapy come together for lymphoma. We knew before that individual agents that make up part of that regimen had activity, but by themselves, each one of them would be effective, but then unfortunately have patients relapse in the end. And it wasn't until we were able to combine those non-overlapping mechanisms that we could actually start to cure a fraction of patients with lymphomas. And I think we're going to see the same thing happen now in the biotech industry as it advances with immuno-oncology. We've got some great innovation going on as well with T cell engagers. So there's now a variety of platforms, checkpoints, the cancer vaccines that we're working on, T cell engagers, and others that we'll learn how to combine as we advance the field. And I think that'll make all the difference in the robust outcomes that we seek for patients with solid cancers.
Yeah. I think we haven't really spoke about you and your background as well, Chris. Can you just share some of the challenges you faced in your career? And then one aspect that I'm always really interested on is that transition from large pharma to smaller biotech. Just walk us through that as well.
Sure. So, I watched the movie Oppenheimer recently and was really struck by the parallels of the Manhattan Project to what we do to advance a new drug. It almost seems impossible with all the things that need to happen that any of us get new drugs approved, and yet I've been involved in a few already in my career, and anticipating many more as we advance the AMP technology. So, what we do is a challenge because we're advancing new technologies that have never been proven yet, and we can't do them by ourselves. So one really striking feature that's again like Oppenheimer is that these all occur in big team science. So you asked about, large pharma versus small pharma or biotech. And I would say that in a way, there's actually not a difference, because the partnerships that are needed to evaluate a new drug in a patient actually always involve a big team of people. So you need a whole network of academic investigators at sites all across a country, or sometimes many countries, to be able to find the patients that have the tumor type for the new drug has promise. And that really means the chief medical officer is kind of like a coach for a sports team, or like a director of a big research program, going for something that's never been done before. You have to do some really careful planning. The larger pharma companies have a diversity of employees with many different skill sets and expertise. So what you'll do in a smaller biotech environment is to build a core team that can execute really well on general activities, but you'll have to be open to partnering with outside colleagues to bring in those skill sets that you can't find in the biotech environment. And you can luckily find great scientific advisors, and even partner with patient advocacy groups and disease foundations that can really help to focus the activities in biotech. So I think, to me, that's the challenge. Is it's certainly not easy to put it all together for the benefit of patients, but it is really rewarding to do that. And the great thing is that biotechs have that unique focus, and really everyone is rallying around the championship of some new technology. I think large pharma can do a good job of this as well, but there's often many competing activities in a larger company, and they're really helpful as you reach late phase, and to take drugs forward that are able to serve patients in multiple indications. There's a real sweet spot there. So I think in my career, I've seen the benefits of both large and small company environments, and enjoyed working in both. I think it's great for young physicians that want to do translation to work actually in both environments as they progress their careers.
Yeah, I couldn't agree with that more. I mean, even from a recruitment standpoint, when I'm looking at profiles and they've worked at big pharma and small biotech, it just elevates, their profile, basically. You talked about those approvals. Can you just give us a little bit more about what it's like, what it feels like? It must be pretty extraordinary feeling.
Yeah, I remember my first one was with a drug for prostate cancer, which was my area of clinical expertise when I was in practice. And that one was from a randomized study in patients who had already been through chemotherapy for the drug abiraterone acetate, which is now Zytiga. And it was developed in a small company and later by Johnson & Johnson. And I remember the colleagues there told me, Chris, you'll never forget the moment that you hear from FDA that your NDA is approved. And it will sort of make its mark on you, just like people remember, when JFK was shot. It's my parents, I think, era. That's what they will remember in their life. And my colleague who told me that was exactly right. That really has always stuck in me. I'll remember the time I saw that email opened up by our regulatory colleagues with that letter from FDA, and we realized that all that work over the years had brought us to be able to actually serve the patients. And the initial approval was in the United States, but now that drug is approved in hundreds of countries around the world. And not a weekend goes by that I don't hear from a family member or a friend of a person who's actually taking that agent as part of their therapy for prostate cancer. It's one of the most common cancers out there, and it's going to always, I think, make an impact on you if you pursue a career in translation like this. That the work that you've done in the past will continue to affect the world around you, and you can see the impact that you've had just every time you go to a barbecue or party, when people hear you're an oncologist and have worked in the field, they'll ask you what you know about their uncle's treatment or their dad or mom's, and that can be very rewarding to realize that sort of impact that you've had. And the flashbulb memories of the milestones like that are really great. You know, they stick with you forever, and are something you can really be proud of.
Yeah, that's what it's all about. That's when it comes down to it. Yes, we're talking fundraising, investors, share price, but like that, at the end of the day, this is what it's all about. And I think it leads really nicely onto what is my last question, which is what advice would you give to aspiring scientists and medical professionals who, like you, are really passionate about making a difference in cancer and immunotherapy?
Yeah, I think it's a fantastic time to be a translational scientist. I think of any kind. You know, you can be a physician, you can be a basic researcher, and what's important is really to follow the science and look for those opportunities to replace the toxic regimens of the past with newer agents. Be they in the immunotherapy field, that's a great one, but there are many other types of therapy that will come along in our careers, a lot of which, you know, you may not have even heard of in your training. You know, don't be afraid to investigate something that has promise that doesn't have a track record yet, because that's how I define opportunity. I do think it's a good piece of advice for young researchers in this field to try to have hands-on experience directly with patients during their training. So for the physicians, I think this is particularly important to have early career responsibility as an attending doctor, to spend some time on the front lines of cancer treatment in my field in oncology. Having that experience being at the bedside will help you to understand how to write a superb clinical trial that can be easily executed if you do go later into the clinical research area. So I think that's really essential. And I really appreciate my time. I had about 5 years as an attending physician at UCSF, treating patients with GU tumors, so prostate, bladder, testicular, and kidney. And that experience really helped me out a lot as I progressed with my career. And I would also tell people to just bear in mind that much more will be discovered as your career progresses. I never would have dreamed of how we are harnessing the immune system now when I was in my training, so that you know, you'll have the chance to make a huge impact on the future of the practice of medicine if you pursue a career in drug development. In cancer immunotherapy in particular, I think that field is very ripe, and there'll be many other fields that we don't even know of today that will come along as your career progresses.
Yeah, amazing. Thank you so much, Chris. Thank you for the time, and it's been an absolute pleasure to have you.
Great, it's great to speak with you, Max. And, look forward to hearing the podcast when that's out there.
Same. I think a lot of people will as well. So look, please everyone like, subscribe to the BioHub, and again, thank you so much, Chris.
Great. Have a great day.